My name is Barry Slaff, and I am a computer and information science PhD student in the Biociphers Lab of Yosef Barash at the University of Pennsylvania, Philadelphia, United States. Today I will walk you through our poster. MAJIQ V3 offers improvements in accuracy, performance, and usability for splicing analysis from RNA sequencing. This work was led by Joseph Eicher, now an MD PhD and resident at the University of Michigan. MAJIQ was released for the first time in 2016. MAJIQ was the first algorithm to detect and quantify complex and de novo splicing events. MAJIQ included the capability to compare splicing between groups of experiments, but the methods were best suited for groups of replicate samples. Realizing that we frequently work with heterogeneous and large groups of samples, we developed MAJIQ V2 to handle such comparisons. In the last year, we released MAJIQ L in preprint. Work led by Song-Woo Han, which extends the original short read algorithms to work with short and long reads together. Previously, we released Moccasin, work led by Caleb Bradens and me, to adjust for known and unknown confounding factors in RNA splicing analyses. So, why do we need a major V3 update to MAJIQ? MAJIQ was originally designed to compare groups of samples, but we envisioned a new clinical pipeline which compares splicing between a single patient and panel of controls. Previous versions of MAJIQ lacked the software features needed for this analysis. Using MAJIQ V3, we have now built this clinical pipeline. You can learn more about the pipeline itself this week during Dina Isakova's MAJIQ Clin Talk with the VAR I Kosi. We also identified opportunities to improve some of MAJIQ's quantification algorithms. Finally, we saw an opportunity to re-implement MAJIQ from the ground up, making optimizations to its data structures and algorithms, which produce faster runtimes compared with previous versions. The decoupled MAJIQ V3 builder is a major improvement in V3, which makes it possible for MAJIQ to efficiently complete the computations needed for the clinical pipeline. Specifically, the clinical pipeline executes many comparisons of one patient against a shared collection of controls, which would have required redundant splicing coverage calculations in previous MAJIQ versions. To avoid this, we decoupled the builder into 3 steps. The first step uses RNA-seq read evidence to update a base splice graph from the transcriptome annotation. The second step combines 2 or more splice graphs into one. The third and final step calculates splicing event coverage from RNA-seq evidence and an input splice graph. Used together, these 3 steps enable efficient quantification of splicing variations for many, perhaps incrementally added, case controls comparisons. In the clinical pipeline, we first update the base splice graph for each case and control RNA-seq experiment. Next, we can combine the control splice graphs into one and quantify splicing event coverage for all cases and controls. Lastly, we can combine each case splice graph separately with the controls and quantify only the additional splicing variations for the particular patient case. We can also incrementally add new cases over time and analyze them against the same controls without redundantly recalculating coverage. These decoupled build steps make it possible to efficiently complete splicing quantifications for many case versus control analyses in a way not possible with previous MAJIQ versions. Another MAJIQ V3 improvement targeting the clinical pipeline is efficient quantile calculation. Briefly, MAJIQ has always modeled percent spliced in, or psi, not as a point number, but as a beta distribution with variance depending on the strength of the RNA-seq read evidence. In order to calculate that splicing in a patient significantly differs from the controls, the clinical pipeline compares extreme quantiles of the patient and control distributions and looks for a psi gap of sufficient size. Hence, MAJIQ V3 must be able to accurately and performantly calculate the quantiles of the patient psi distribution. MAJIQ V3 includes algorithmic updates which result in a relatively accurate and performant calculation. Another MAJIQ V3 improvement is in retained intron quantifications. In addition to spliced junctions between exons, MAJIQ has always included retained introns as splicing variations of interest. MAJIQ V3 improves the accuracy of retained intron quantifications with an algorithmic update that avoids counting read coverage from an exon towards introns of an overlapping gene. We note from our performance evaluations that this was rarely an issue, but still something we wanted to improve. One more improvement in MAJIQ V3 is the direct integration of Moccasin, our method which adjusts for confounding factors. Previously, Moccasin was released as separate scripts together with MAJIQ V2. MAJIQ V3 integrates Moccasin directly, taking advantage of the improved V3 data structures for a more efficient implementation and decoupling the Moccasin steps so models of confounding variation may be calculated once and then reused across executions. Our performance evaluations show strong concordance between the psi and delta psi quantifications in MAJIQ V2 and V3. The psi values show strong correlations across splicing event types and coverage levels. MAJIQ V3 also shows dramatic runtime improvements over MAJIQ V2 while retaining a modest memory footprint. These improvements are aided by the new MAJIQ V3 implementation, which includes algorithmic improvements and contiguous data structures such as Dask arrays. These arrays can be manipulated efficiently for linear algebra operations and are relatively performant in disk I/O operations. MAJIQ V3 is not yet available to download. Please check our website, majiq.biocyphers.org, for upcoming release news. Thank you for checking out the MAJIQ V3 poster, and please be sure to check out Dina's MAJIQ Clintalk with the var I COSI as well. I hope you enjoy ISMB Montreal.